Hi, welcome back. This is part 2 of chapter 25. I wanted to give you a quick little overview kind of what we're doing, and then we'll move through each section in a little bit more detail. So remember, afferent arteriole is delivering blood plasma, right? And it drops through the glomerular capillaries and it's collected in this bowl, which is the glomerular capsule. And now we've created filtrate. So remember, the smaller stuff becomes the filtrate. This is going to be water, and this is going to be solutes. And then the large stuff gets kicked out to the efferent, so that's the proteins like albumin, red blood cells, things like that, right? They're going to go efferent arteriole. So all the small stuff drops through. This is our filtrate. And then we come down here, we're traveling, the filtrate's traveling in the tube, and here at tubular reabsorption, we just say, okay, we need to keep all of this. We need sodium, we need potassium, we need calcium. Everything that we're going to keep, we're going to reabsorb into the capillary. And remember, this capillary bed is eventually going to go to a venule and then eventually inferior vena cava. So you're putting it back into the bloodstream. And then at the end here, we have tubular secretion. Oh, wait, we have too much of this. We can go ahead and get rid of it. Maybe we have too many hydrogen ions, right? Maybe our blood is too acidic and we need to get rid of some of those. So anything that we don't need, it's kind of our last ditch effort to dump it into the tube system so that it can become urine and that we can get rid of it as waste. So we filter, we reabsorb, and then we secrete. So we have three processes going on. We have the first is glomerular filtration, right? And it produces our filtrate. Then we have tubular reabsorption, which returns all the glucose, amino acids, 99% of the water, salts, and the components of blood that we just got from the filtrate. 99% of it gets put back into the bloodstream via the capillaries. And then we have this third part, which is tubular secretion. So basically, if we want to selectively add anything to our urine or mark it as waste product. Let's look at this part first. We're going to look at the glomerular capsule and the glomerular capillaries in more detail. Here's a nice image of your renal corpuscle. Remember, your renal corpuscle has to have two components or two parts. It has the capillaries, so I'm going to abbreviate here, glomerular capillaries, that's this part. And then it has the glomerular capsule, and that is this part. So remember, the afferent arteriole is delivering blood plasma, and then it all gets filtered through this glomerular capillary. Remember those fenestrated capillaries? And what gets dropped into the glomerular capsule, it's going to now be called filtrate. And what's going to be in this filtrate? We saw from the last slide, we're going to have glucose, you could have amino acids, you could have water, you could have solutes like salt, right, ions. And that filtrate is going to travel from the glomerular capsule into the proximal convoluted tubule. Kind of just the words of what we just saw. The fenestrated capillaries allow for specialized filtration, right? These have holes. They allow for the passage of water and solutes. Afferent arteriole enters the glomerulus capillaries and the efferent arteriole exits. The blood pressure in the glomerulus is high because the afferent arterioles are larger than the exiting efferent arterioles. Arterioles are high resistant vessels. So let's look at the image again. What do you notice about the afferent versus the efferent? Do you see how the afferent is much larger than the efferent? So blood pressure going in is high because it cannot leave as fast. So we're going to have a little bit higher pressure here in the glomerulus. Why would we want that? If we have higher pressure here and we have lower pressure here, does that allow for more filtrate? Hopefully you're saying yes, we know this. We remember hydrostatic and osmotic pressure. Something that I think is important to point out about this glomerular filtration is that it's a passive, non-selective process. So remember, passive, no ATP required, right? No metabolic energy. All we're doing is moving the blood plasma through the capillary, right? And then it forms the filtrate. So they want you to realize that this is just passively happening, but we also have to think about the hydrostatic forces that are at work here to move the solutes through the filtration membrane. So it's the hydrostatic pressure that's greater in the glomerular capillaries that allows for those small solutes to move through the capillary bed into the glomerular capsule and eventually become filtrate. We get to talk about hydrostatic pressure again. Yay! I know you're saying boo, but this should be easy now after we've done it once, hopefully. So we have the hydrostatic pressure inside the glomerulus here. So it's pushing down at about 55 millimeters of mercury. The hydrostatic pressure that's pushing back from any filtrate that might be here is about 15. You also have this osmotic pressure. This is the pull from the proteins that are in the blood. It's pulling water towards the proteins. But if you look at this, what's the overall? So we have 55 pressing down from the glomerulus, and we have 45 pressing back from the fluid that's in the glomerular capsule. That still gives us a net of 10. So what's the net pressure? Where are we moving? We're moving fluid from the glomerulus capillary into the glomerular capsule. So we're moving from blood to filtrate. We have to talk about something called glomerular filtration rate, and then we'll go back and talk about why the rate or the pressure is important as the blood is entering the afferent arteriole. So they're going to start abbreviating it as GFR, glomerular filtration rate, right? That's the volume of filtrate formed per minute by all of the glomeruli. So not just one glomerulus or capillary tuft, but all of them, right? So it's the amount of filtrate that you're forming each minute. A constant glomerular filtration rate is important because reabsorption of water and solutes depends on how quickly the filtrate flows. We talked about this with digestion. You do not want food just whipping through your intestines, right? If the food doesn't have a long enough time to sit there, you cannot reabsorb the nutrients from the chyme that's in your small intestines. So we want kind of a slow and steady, right? If you're trying to reabsorb and detect things like glucose and solutes and amino acids, you want that to be moving slowly through the kidney. So glomerular filtration rate should be constant if you want to have optimal reabsorption of nutrients. And the normal is about 120 to 125. And something that I'll mention is if you've ever had a complete blood work done, if they've looked at glomerular filtration rate as well, you'll see that there is an African American glomerular filtration rate, which is higher than Caucasian glomerular filtration rate. So back to this image, can you see why the blood pressure in the afferent arteriole would make a difference on the amount of filtrate that you're making here? So what happens if your blood pressure is too high? If your hydrostatic glomerular capillary rate is too high, if your blood pressure is too high, could it damage the capillary bed? What happens if it blows a hole in your glomerulus capillary? What happens now? What's supposed to be kicked out? Proteins like red blood cells? So what happens if your blood pressure is too high? You destroy a section of your capillary and now you have red blood cells in your filtrate. That's probably not good, right? You've also destroyed part of the capillary that should be filtering out all of the large proteins, and that particular nephron's not going to probably work as well. Now, yes, we have millions, but if you have consistent, constant, chronic high blood pressure, that's going to affect the hydrostatic pressure in your uh, glomerulus capillaries, and that's going to cause problems with your kidney filtration rate. Thankfully, we have a way to regulate glomerular filtration. Glomerular filtration is tightly controlled by intrinsic mechanisms, so inside the kidney, and extrinsic mechanisms outside the kidney. So we'll talk, although this is triggered inside, it's still extrinsic because it goes, remember, renin goes to angiotensin 2, which then goes to aldosterone, which is outside at the adrenal gland. So we have intrinsic controls or regulations. So this is inside the kidneys. The smooth muscle in the afferent arterioles can contract or dilate, constrict or dilate. So this is going to seem a little strange to you. I'm going to show you on the next image because normally, what do we think? With systemic, if the blood pressure is high, we do dilation, right? We have high blood pressure, we're going to do dilation, and what's going to happen to the blood pressure? It's going to drop. This is different when we're talking inside the kidneys and we're talking about this afferent arteriole. It's the opposite. So if the blood pressure is high, we're going to constrict the afferent arteriole. Basically what we're doing is limiting the amount of blood that can get into the glomerular capsule. If the blood pressure is low, we're going to open up the afferent, open wide. We need more blood flow. Because what are we trying to do? We're trying to keep that glomerular filtration rate constant, right? Maybe it'll help to look at this image. So remember, here's our afferent arteriole. Here are granular cells. And we said we can do vasoconstriction or vasodilation. Remember, the end goal here is glomerular filtration rate staying constant, that 120 mLs per minute, right? So we want to keep this glomerular filtration rate constant. So if your blood pressure is too high, we're going to constrict. We're going to limit the blood flow to try to keep this number constant. If our blood pressure is too low, then we're going to open this up. Let's get more blood going to the glomerular capsule or the glomerular capillaries, right? So if the blood is trickling, into the capillary, we want to open it up and let more blood. If the blood is rushing like a fire hose into the capillary, well, we want to slow that down. So it's kind of a different perspective, right? So I would just think if the blood pressure in the afferent arteriole is high, we're going to do constriction to kind of slow it down, turn the spigot down, right? So we don't have as much water going into the capillary. And that's going to help keep that glomerular filtration rate constant. The second intrinsic control, or the inside control by the kidneys to regulate glomerular filtration rate, is by those maculodensa cells. So let's see if you can follow this, and then we'll look at the image. They're saying if your glomerular filtration rate increases, so if your GFR is high, that means that your filtrate increases, the amount of filtrate increases. You just have blood rushing through the capillary, more filtrate. If your glomerular filtration rate is too high, you're going to have decreased time and ability to reabsorb. That's going to cause you to have high levels of sodium chloride in the filtrate. What should have happened to that sodium chloride? We should have reabsorbed it, right? So if your sodium chloride level is really high in the filtrate, that is basically telling you that your glomerular filtration rate is too high. It needs to slow down. So remember how we said the maculodensa cells could talk to the granular cells? Well, that's exactly what happens. The maculodensa cells can talk to the granular cells and cause the afferent arterial constriction that we just talked about. This can lower the glomerular filtration rate and allow more time for the sodium chloride reabsorption. So then the macula densa cells, if this kicks in, if the afferent arterioles are constricted, if your glomerular filtration rate goes down, if you have more time to reabsorb sodium chloride, then your sodium chloride concentration should go down, and your macula densa cells will see that and think everything's fine. So the macula densa cells here are going to be monitoring the filtrate in the tube system. So they're monitoring the filtrate for increased sodium chloride. If there's too much sodium chloride, if the sodium chloride is high, that must mean the filtrate rate is high and we have decreased absorption. So the macula densa cells can talk to the granular cells and tell them do constriction. Basically, they're saying the blood pressure is too high, the glomerular filtration rate is too high, my filtrate flow is too high, my sodium chloride is too high, I'm not doing absorption efficiently. So that triggers the granular cells to cause vasoconstriction of the afferent arteriole, to slow down the blood flow, to slow down the glomerular filtration rate so that the reabsorption of the sodium has enough time to actually take place. So what are some of the outside regulatory mechanisms that are happening to affect the glomerular filtration rate? Well, you should automatically think sympathetic nervous system. Remember, that's our fight or flight. So what's going to happen with sympathetic nervous system? We're going to increase blood pressure, right? This is systemic blood pressure. We're going to increase systemic blood pressure, increase heart rate, breathing, right? This is triggered by epinephrine and norepinephrine. Those are our adrenaline hormones that were released from the adrenal gland, specifically the adrenal medulla. So this is kind of showing you what's happening both systemically and at the kidney level. So systemic vasoconstriction. So we're going to constrict the blood vessels. And if we constrict systemic blood vessels, that increases blood pressure systemically, right? Why do we need this increased blood pressure? We want our blood moving around faster. We want oxygen being delivered to our muscles and our brain faster, right? So that we can fight or flight. But what's going to happen at the kidney level? So this is at the kidney level. We're going to constrict the afferent arteriole. We're going to turn the tap off, right? We're going to decrease blood flow and decrease blood pressure to the kidney. What's going to happen to the glomerular filtration rate? And then what's going to happen eventually to your urine output? So there's another extrinsic control. It's the Reninangiotensin mechanism. So remember, eventually this is going to trigger aldosterone and eventually trigger sodium reabsorption. That causes water to be reabsorbed, which causes increased blood volume, which causes increased blood pressure. This is the main mechanism that we have for increasing our blood pressure, and it can be triggered by those granular cells. So the granular cells are the ones that actually release renin. Remember, these are those mechanoreceptors, and they're monitoring the afferent arteriole blood pressure. So the granular cells are monitoring the afferent arterial pressure, and they can trigger the release of renin to increase blood pressure. Because remember, these granular cells are trying to keep a stable glomerular filtration rate by monitoring and changing the afferent arterioles that lead into the glomerulus. But there are multiple actions that can cause renin to be released. So the granular cells can release renin. The granular cells can be told by the sympathetic nervous system to release renin. The macula densa cells can tell the granular cells to release renin. And then the stretch. So if we have reduced stretch of the granular cells, Remember, they're the mechanoreceptors that I just talked about. So if you have reduced stretch, that means the blood pressure is low. So then the granular cells are going to release renin. So it's all kind of tied to these granular cells because they're the ones that actually release the renin. But I want you to see that there's multiple ways that renin can be turned on. This is a nice image showing you the systemic blood pressure. If it's decreased, what's going to happen? So here's intrinsically, here's extrinsically. So intrinsically, inside the kidneys, what can happen is if the blood pressure is low, the glomerular filtration rate will be low. And the smooth muscle mechanoreceptors are in the afferent arteriole are going to detect that. And it's going to cause vasodilation of the afferent arterioles. Basically turn up the fluid volume, move more fluid through the glomerulus so that I can increase my glomerular filtration rate. Also, if your glomerular filtration rate is low, that's going to cause issues with your sodium chloride concentration, which your macula cells are going to recognize, and they're also going to tell the cells near the afferent arteriole, hey, you guys need to do vasodilation. So you have both vasodilation of the smooth muscle at the afferent arterioles, and you have the macula densa, both triggering the same event, which is going to cause your glomerular filtration rate to go up. Outside of the kidneys, so this is extrinsic, if the kidneys, the granular cells of the kidneys release renin, it turns into angiotensin, it goes to the adrenal cortex and stimulates the secretion of aldosterone. Then we have reabsorption of sodium, reabsorption of water, and we have an increase in our systemic blood pressure. What else can we do? We can do vasoconstriction of our systemic arterioles to increase the resistance, right, and increase our blood pressure. So hopefully that made a little bit more sense. They do talk about here the sympathetic nervous system and how it can also independently stimulate the granular cells to release renin. Another clinical homeostatic imbalance, you can have anuria. So basically A means not, right? Abnormally low urine output. It could mean a number of things. It could mean that something's wrong with your blood pressure. Your blood pressure is too low to cause filtration, so you're not able to push blood through the capillaries, the glomerulus, right? It could be renal failure. It could be some sort of infection. It could be transfusion reactions. Remember, if you have agglutination, if you have antibodies that are binding and kind of gumming up and causing the clumping or agglutination, that all has to be cleared through the kidneys. So imagine now that all of those antibodies stuck to all of those antigens are now clogging up the kidneys. It could also cause abnormally low urine output. So now we're on to step 2, which is tubular reabsorption. So once we filter everything through, so we've got our little cup and we've got our capillaries and we have blood coming in, and we're going to drop everything through to the glomerular capsule, and now we have our filtrate, right? The very next step is this tubular reabsorption. Remember, we said about 99% of what we filtered through, we're actually going to reabsorb. So tubular reabsorption quickly reclaims most of the tubular contents and returns them to the blood. All organic nutrients are reabsorbed. When I say organic, you're hopefully thinking of anything that has carbon, right? So you're thinking about glucose and you're thinking about amino acids. So water and ions are reabsorbed. And then we have active and passive tubular reabsorption. So remember how I said the kidneys filtering your blood is about 20-25% of your oxygen demands? You do have some active transport, which is going to require ATP. Let's just look at the figure. Don't worry too much about the words up here. Just kind of follow me. We have the filtrate. So we're in the proximal convoluted tubule and we're doing reabsorption, right? We have water and we have solutes. The water is flowing passively either between the cells, pericellular, or through the cells, transcellular. So we have passive transport of water going into the peritubular capillary, and this should make sense because peritubular wraps around the proximal and the distal convoluted tubule. These are the cortical nephron, right? I know if I see peritubular, I'm talking about a cortical nephron. And then we have solutes. Remember, solutes are usually charged. If you're a charged particle and you're trying to cross a membrane, usually you have to do active transport, right? Protein-assisted active transport. See, it all comes back. So you can see here, they can do paracellular. They can go between some of these ion channels, and then some of them are going to go directly through and do transcellular transport. So I just want you to see there's kind of a mix here. There's some active transport, there's some passive transport, but during the tubular reabsorption, you're picking up almost everything and dumping it back into the bloodstream. We need to focus on the fact that sodium is the most abundant cation in the filtrate. Remember that sodium is really important for setting up that resting membrane potential, right? So sodium reabsorption also provides the energy and the means for reabsorbing most other substances. So what's really important here is that sodium is co-transporting many other types of solutes, like glucose and amino acids and vitamins, other cations, other positively charged. Remember, like we have sodium and potassium that are transported together when we talked about ATPase pumps. So sodium is the most abundant positively charged ion in the filtrate, and you need sodium to co-transport many of the necessary molecules that we need to reabsorb. So again, this slide is just showing you what's happening to some of these ions and solutes in the filtrate. So sodium is co-transported with a lot of ions, including glucose and amino acids and some other vitamins. So basically, to get these guys across, you have to have enough sodium. They have to be co-transported or at the same time. Water can travel through the kidney tubule into the peritubular capillary through passive either ion channels, right? Or in some cases, if we have lipids, they can just pass right through the membrane. So if you're lipid soluble, remember this is a phospholipid bilayer, so they're both made of the same material, so the lipids can pass through. And what would be some of those if you wanted to reabsorb some vitamins? Remember, some vitamins are lipid soluble and some are water soluble. And then we have some other ions that sometimes they have channel junctions and they can get through the proximal convoluted tubule into the peritubular capillaries through some sort of junction. The movement of sodium and other solutes creates a gradient for water. Remember osmosis, water goes wherever it can dilute out the solutes. In this case, they're talking about sodium ions. So water is reabsorbed by osmosis, and we do have some pores called aquaporins in the cells. So those can specifically allow water to be transported. But the real point of this slide is to talk about kind of these carriers. So if you had a glucose carrier, let's say a protein that's going to allow glucose to bind and move through the cell and put back into the capillary. If that carrier, if that protein is full or saturated, the excess is going to go to urine. So basically, we're talking here about proximal convoluted tubule and reabsorption. So we're saying if we're all full, if the proteins are all filled with glucose, where's the excess glucose going to go? It's going to go distal convoluted tubule, it's going to go collecting duct, and eventually it's going to go through the kidneys and be processed as urine. So if somebody's a diabetic, they have low or no insulin response. Remember, insulin is what tells the cells to take in glucose. So if you have no insulin or low insulin, you have high glucose in your bloodstream because the cells aren't taking it up, right? So high blood glucose levels. Once the carriers are saturated for glucose, that excess has to be converted into urine or gets put in the tube to become urine. So this is why somebody that's diabetic, one of the things that we talked about is that their urine smells really sweet because they have tons of glucose that cannot be reabsorbed, so it has to be released in their urine. If we wanted to do a summary of tubular reabsorption, what's happening? This is happening at the proximal convoluted tubule. This is where the majority of reabsorption happens. We're talking about the majority of the nutrients, right? So glucose and amino acid, all of these should be reabsorbed. We should reabsorb a lot of sodium and a lot of water because they're co-transported, right? To reabsorb these guys, we have to have lots of sodium, lots of ions, almost all of the uric acid, and about half of the urea. And we'll talk about what's going to happen to the rest of this later. So urea is from protein breakdown, and uric acid is from nucleic breakdown. So nucleic acids and proteins into amino acids and turns into urea. Even though the majority of tubular reabsorption happens in the proximal convoluted tubule, we also have to talk about some other portions of tube, like the nephron loop. This is going to reabsorb water, right? So we're going to look at this in a little bit more detail. And also, we have tubular reabsorption happening in the distal convoluted tubule and the collecting duct. So really, tubular reabsorption happens in the proximal, in the loop of Henle, in the convoluted tubule, and the collecting duct. Reabsorption here, though, is usually hormonally regulated. So the distal convoluted tubule and the collecting duct have receptors for different hormones that can increase water absorption, can increase sodium reabsorption, right? Can increase calcium reabsorption. So these are more hormonally controlled tubular reabsorption. Step 3, we have tubular secretion. So basically, after we do the filtration, then we do the reabsorption, then we can kind of do last-minute tubular secretion. It does occur in the proximal convoluted tubule. It can also occur in the distal convoluted tubule and the collecting duct. But these, like I said, are more hormonally regulated. Substances are moved from peritubular capillaries into the filtrate. Eliminates any undesirable substances. If we want to get rid of some of the urea or uric acid, any of the drugs that might be in the system or metabolites from the breakdown of the drugs, any excess potassium we need to get rid of, also any extra hydrogen ions or bicarbonate ions that we need to get rid of. So this should clue you in that this has something to do with that bicarbonate buffer system that we talked about in respiratory, right? This is a nice image because it shows you that both reabsorption and secretion are happening in the same section of tube. So this is the proximal convoluted tubule, and we have reabsorption of water, of ions, bicarbonate, sodium, amino acids, glucose, and other nutrients. We're reabsorbing these into the bloodstream. Remember, the peritubular capillaries are wrapped around. So we're putting this back into our blood. We're reabsorbing. But what are we putting in the tube system as waste? What are we adding to the filtrate? So secretion is adding to the filtrate. Maybe some extra hydrogen ions we don't need, maybe some metabolites from drugs that we've broken down. And then when we get to the loop here, loop of Henle or the nephron loop, we can reabsorb water. We can also put more things into the tube system, into the filtrate that we want to get rid of. Maybe we don't need all this urea, we want to dump it into urine, right? So the loop is also absorbing and secreting. And then we get over here to the distal convoluted tubule. It's also reabsorbing. It's also secreting. Remember, again, the peritubular capillaries wrapped around here as well. So we're going to reabsorb, basically directed by hormones. We're going to reabsorb sodium if we need more, calcium if we need more, but we could also get rid of, if we have too much, potassium. So I think this is a really good image to show you that it's not clear cut, that absorption and secretion are happening in all sections of the tube. It's not just one or the other, it's both.